So I'll go ahead and get started with kind of some of the basics of the technique. And I think you guys have the slides in your little binder if you want to sort of follow along and take some notes in there. So I guess the fundamental questions are why do we want to do phosphoflow? So why phospho and why flow cytometry is the two parts to this equation. The first part of why you want to do signaling, at least from my angle, is that most of the events that a lot of people are used to maybe measuring, things like cell differentiation or protein secretion, gene transcription, these things tend to take a little bit more time. You might be on the scale of hours to days or weeks, whereas phosphorylation is really kind of that first fundamental event that happens. And so there's not a lot of things that kind of happen upstream of signaling. Signaling is really the first thing that you can measure. And so it's a really good readout of how the cell functions or how the disease might be modulating a particular cell. So you can have things like cytokine signaling, which we'll do in the class this afternoon, where a cytokine binds, leads to activation of STAT transcription factors, which we can measure by phosphoflow, and you get some kind of transcription. You can have MAP kinase activation, whatever you might be interested in measuring, and the number of antibodies keeps increasing that are available now for the pathways that you might be interested in. So flow versus kind of the traditional methods, why flow? So traditionally, you kind of look at a blot and you just get some kind of number for a population, whereas with flow, you can get each individual cell running through there, you get data for that cell. Traditionally, you have kind of single parameter data, whereas flow, as everybody's talking about, I used to put 10 to 13 up there as kind of like, ooh, 10 to 13, but apparently that's where everybody is now, so I should move that up to 25 or something. So typically with the biochemistry methods, you're stuck a little bit in kind of the more homogeneous cell types too, so you can't tease apart the different subsets, whereas with flow, again, using multi-parameter nature, you can pick apart the different cell subsets, so you can work in heterogeneous populations like primary cells, which most of you are interested in. You typically need a lot of cells to be able to do this kind of biochemical techniques, whereas with flow, as you know, you can get into these rare subsets. You can go in dendritic cells, stem cells, whatever, antigen-specific T cells, so you can dream it up, I think, with flow and really be able to analyze signaling in those subsets that you could really never actually analyze the signaling for with the biochemistry. It's just not feasible to do. Really, you don't want to be running a ton of blots. I don't have to say any more than that. With flow, you really can run a lot of samples by flow. There's a lot of auto samplers and things too, for these instruments to really let you run hundreds of samples pretty easily. And this is a great advantage too, to really be able to look at time courses, dose responses, disease courses over time, all these kinds of things that I think by normal methods you're kind of constrained by what can I put on a blot. I'm going to put my 10 lanes or something like that, and you're not going to be thinking, I'm going to run 15 blots to get all this data. So again, the traditional techniques, you basically take a bunch of cells, you mash them up, and you get that one value for that population. So you just get one average. You really have no sense of what the distribution is. Whereas when you want to run something by flow, again, as each cell goes through, you get a value for each individual cell, and so you can start to see that distribution. You can start to see, is there some heterogeneity in that sample. And again, you can measure a lot of parameters per cell. So now you can have 10 plus parameters per cell, so you can have cell number 3 here, and then you can have all your different fluorescent parameters with your different readouts or your different mass tagged antibodies now too, since multiple people here are acquiring cytophs, and no pressure in Africa. Ron will help you out. It'll work, right? Yeah, I'm sure he would. So you can go 30 whatever out here and get all those individual measurements now per cell. And what that lets you do is to move into, again, these heterogeneous populations of cells. So you can imagine kind of now making panels where you have surface markers that might identify your different cell subsets, and then because we're talking about signaling in this class, you might throw in some antibodies against different phosphoproteins kind of mixed in there. So these different antibodies against the surface markers will help you identify your different cell subsets, the yellow ones, the blue ones, the red ones. And then you can look at, 3 different phosphoproteins or something in each cell subset. So now from one sample, you're basically getting kind of 9 data points, 3 cell subsets, and 3 signaling protein measurements. And again, you can pick out those rare cell types where, you can have those antibodies that, as you know, and you've stained before, you can have your 1 out of 100, 1 out of 1,000 kind of cell population that you can identify with those particular antibodies. And again, that's really kind of the magic for us that's let us look at a lot of novel areas, basically in the disease-specific cells, or just those rare cell subsets that you just couldn't kind of get to before, regulatory T cells or subsets, really kind of esoteric subsets of dendritic cells, monocytes, and these other populations that might be more rare in a particular tissue. So because we're going to be talking about it a fair amount, and because the experiments are going to kind of rely on cytokine signaling, I wanted to touch a little bit just to get everybody on pace a little bit with cytokine signaling and just the real basics of it. So most cytokine signaling happens when you add, or there's a cytokine secreted on the outside of the cells. This dimerizes typically cytokine receptors that tend to be a lot of the time heterodimers, sometimes homodimers. You have the JAK kinases, which are docked to the intracellular part of the cytokine receptor. And then that activation typically leads to the docking of the STAT transcription factors and their phosphorylation. And the STAT transcription factor is basically the end game for this pathway. That goes into the nucleus and causes transcription. So overall, the signaling pathway is kind of a simple signaling pathway on the scale of things, I think, and lets you analyze it kind of quite easily too, and there's not too many things that confound this, although what leads to actual STAT phosphorylation now, I think a lot of people are showing that other things, especially in cancer, can feed into this and lead to kind of constitutive activation of the STATs. So overall, it's pretty simple: cytokine receptor, JAKs, and STATs. So the actual technique that we use most of the time is also pretty straightforward. So the first thing that we do is fix the cells with formaldehyde, and we'll kind of go backwards and talk about kind of how we got to this technique. So we fix the cells with formaldehyde, and that basically stops all the signaling, kills the cells so nothing else can happen. And this is important, obviously, for cell signaling. You don't want to have your cells continue to signal as you're trying to work them up and analyze what's going on inside them. So you need something that's very rapid. And a lot of signaling pathways really happen on the scale of seconds to minutes. So if you sit there and you just spin it down and you try to cool it down or something like that, you really might just completely miss that signaling pathway. So the fixation is really important to do as rapidly as possible, particularly for sort of a lot of really upstream signaling pathways or signaling proteins. The next thing that we typically do, although there's different methods, is permeabilize with methanol. So you need some way to basically get your antibodies inside your cell, right? So the cell is fixed. Fixation on its own maybe permeabilizes a little bit, but not a whole lot. So you need some way to get your antibodies in, and so we typically permeabilize with methanol, and then we come in with our antibodies against our phosphoproteins, and these are typically fluorescently labeled or now labeled with mass tags to measure on the cytoph. And then we run them on the cytometer and we get our signal. So what does it kind of typically look like? The typical kind of measurement, if you have the autofluorescence of a cell down here kind of in a dotted line, your typical signal, you have a little bit of background basically on your cells, as you typically do in kind of any experiment, where you have some antibody binding to either stuff inside the cell or things on the surface of the cell. And I would say one of the changes for people that kind of haven't done phosphoflow and people that have, probably the rude awakening is that a lot of things stick to stuff inside the cell, including your surface marker antibodies once the cells have been permeabilized. So most of these surface marker antibodies have not been optimized for intracellular staining. So when you throw that antibody in there, all of a sudden it's seeing 30,000 other proteins that it normally doesn't see, and that causes a lot of problems for these things, and they're just not necessarily specific. So surface marker antibody titration and kind of optimization is probably actually the most important part of phosphoflow. I think the phosphoproteins are kind of straightforward on the scale of things. It's really all about getting those surface markers actually to give you the correct subset, as I think someone pointed out too, that they had tried, and that was the biggest problem, that they weren't seeing kind of the right subsets. So then going back kind of the signaling molecules, if you had a good robust induction or you had a high level of phosphorylation in, say, a disease, you should see kind of a higher level of fluorescence, that correlates to the binding of that antibody to the inside of the cell. So you have this nice kind of increase in signal. So just to kind of correlate it back to what a Western might look like, you can see, in essence, a band in a Western blot kind of correlates to a shift in your phosphoflow. So you might have low or just kind of autofluorescent signal. You induce a signaling pathway here, interferon gamma inducing STAT1, and you see an increase in that signal. And so an increase in the signal correlates to an increase in the phosphoprotein level. You can measure intermediate levels. So here you have a couple stems that induce something intermediate, and then a high level here. So you can see kind of those intermediate and high levels. And what we typically do is kind of a simple metric, although there's all kinds of metrics, but you'll hear this kind of used a lot. What we started off using was what we call the fold change. That's basically just the fluorescence after stimulation divided by the fluorescence before stimulation. And so this is a really simple metric. You might have, say, 100 units here and 10 units there, and it would basically be a 10-fold change. So you'll hear us say that a lot. It's kind of some fold change. You might also have some kind of derivation of the fold change, a log conversion of the fold change or something like that to help you visualize it a little bit better. But in essence, this is something that we use most of the time as some version of kind of a fold change. So again, phosphoflow is a quantitative single-cell analysis technique for intracellular signaling. So how did we get to this optimal protocol? So we first tried out, this is kind of from, it's almost 10 years. Whew, scary. So we've been doing this for a while now, as you can see. And we basically did some different methods where we did no fixative or we added a fixative. And then we tried different perm reagents, some alcohols or organic solvents, and then also some detergents. And just to kind of jump to the point, you can see that the fix followed by methanol tended to kind of be the best one across the board. Looking at a couple of the MAP kinases and also a couple of the STAT proteins. Depending on the pathway that you're looking at, you can really still optimize this always. So I would never say that this method is the best method to use for everything, because you really can find ones that are better for your particular pathway. So I always say if there's really only kind of one thing that you want to look at, try different levels of fix, try different permeabilization reagents. You can really dial into certain pathways. This is the one that we kind of use that's the most generic, I would say, and tends to kind of work most of the time. And at least I haven't seen it where this method hasn't worked. It might not have worked as well, but basically this pretty much always works to some extent if you can see some other method work. The other method might be a little bit better, but again, the kind of the fixed methanol is pretty much always going to give you something. Again, might just not be as good of a signal to noise. So formaldehyde fix followed by methanol perm has kind of become our standard method. For people using the BD buffers, this is the kind of I forget what they call their fix buffer.
Sorry, what percentage of the?
We use 1.6% normally. Yeah. Yeah, what's the BD fix? I'm sure someone's bought it. I always forget that. Is it fixed perm though?
Well, we use that for ICO.
So, yeah, they use. I think they also have one that's the PhosFlow like fix buffer or something like that. I always get confused. As you can tell, we don't use that, but you can just buy formaldehyde and do the job too. The methanol is Perm Buffer 3, so if anyone's interested, that's what they sell is Perm Buffer 3, is basically almost all methanol. And so that's kind of the equivalent of basically using methanol if you'd rather buy a kind of a pre-made buffer from BD. So optimizing kind of this technique, and this I'll just kind of use as a way to point out how you can still make this method better. So the fix time, we kind of did something for fixing for even just a minute, 10 minutes, or 30 minutes. And you can see looking at, say, the MAP kinase here versus the STAT protein, you can actually see that the MAP kinase staining got a little bit better. So this is an unstimulated versus stimulated sample. And you can see that that actually got a little bit better. Sort of a longer fix actually improved that slightly. Whereas with the stat, you actually got a slight decrease in the signal overall there. And this is just something that I'll point out is very common. So typically if you optimize for one phosphoprotein, you're going to lose a little bit on the other side, on some other protein that you want to look at. So that's also where kind of this fixed methanol combination has really kind of become our standard, because although you might improve something, with kind of optimizing it, you'll probably sort of, make something else worse at the same time. So it's a little bit of one of those things. You want to look at all kinds of signaling proteins. If someone has a cytoph and they want to do 10, then I would say probably, fixed methanol is a good way to do it. If you're really interested in, say, P38 signaling in your particular disease, then I would say it's probably worth the while to kind of see, how long should I fix, or how hard should I fix, or should I try some different permeabilization reagents to really get an optimal signal there. So it's a little bit of balance. Do I want to look at a lot of things, or do I really want to look at one? If you really want to look at one, I'd really, really, emphasize that you should try to kind of optimize that, because you can make your signal to noise go from 3x to 6x even sometimes, or something like that, by just optimizing out that technique. The fixed temperature, room temp versus 37 degrees, we really haven't seen much difference there. Again, certain pathways might benefit from that, but for the most part, we tend to just put the fix in and then just leave the cells out at room temp, and that makes it quite convenient. You don't have to throw it back in an incubator or a water bath. The methanol permeabilization time, you can see a pretty big jump here from 10 minutes to a week to 5 weeks. And this is actually stored at -20. It's even better at -80. But you can see here at -20, there's actually a little bit of decrease for some of the signaling pathways over time. If you store it at -80 in methanol, you lose a little bit, but not a lot. So it's actually quite robust to leave your sample stored at -80 in methanol for long periods of time. And we've done this actually to just accumulate samples over time for kind of longitudinal studies where we have, let's say, mouse samples or something that we do a longer, months kind of thing. And we'll get the different samples at different time points, get them into methanol, and we'll store them in there for a few months. Then you can pull them out, all at one time point and do the staining, at that later time point. So it's really advantageous for having your stain be consistent, instead of having kind of a stain at all these different days where you might just have some wobble based on, how accurate you are every time you stain, so, and how your instrument is tuned and things like that. The staining time, 15, 30, 60 minutes. I would say for the most part, we stain a lot at half an hour, but you can definitely improve things too for certain antibodies, especially now with some of the PE conjugates and some of these other bigger fluorophores. Sometimes they're staining for longer periods, so doing an hour or maybe even 2 hours can be advantageous. So if you do some of these other fluorophores now, outside of kind of the small molecule ones, the antibodies do tend to take a little bit longer to kind of diffuse into the cell, it seems like. So I would recommend going a little bit longer for some of those larger conjugates there. It's worthwhile too to kind of compare some of your fluorophores and just see kind of which ones are brightest. So for the most part, your kind of further red dyes like Alexa 647 versus the kind of Alexa 488. You tend to get a better signal with the 647 relative to the 488, but certain antibodies, you'll find kind of the ERK and the STAT6, for instance, when we did this, will actually be quite comparable between those. So, if you have the ability to try a couple of different colors for things, because, you might find really that, okay, I can put ERK on either color, but, one of them, STAT5 or something I really want on 647, so it leaves you open, for being able to do that. And so just ask the vendors for samples. That's one thing I would always recommend too. Don't just buy a $300 vial. Ask them for a little bit first. I found most of the time they'll give you a little bit. I don't know what Ron will tell you, but you gotta try it. You gotta try it somehow. And so I think a lot of these things, lets you kind of test it out and see which one might work in the best format for you. So again, the technique is quite quantitative. This is just to show a Western blot, and then phosphoflow with STAT1 on two different colors, 647 and 488, and just kind of a dose response curve. And you can see those curves correlate really nicely. And again, that's important, so people that want to do drug activity studies or just any kind of cytokine dose response or agonist, antagonist studies, you can do it with phosphoflow, and it's quite quantitative, and we've done this over and over again, so it works quite well. You can also do obviously multiple phosphoproteins at the same time, so it's an experiment that we used to do, and now we've kind of gone past this. This was the old days of the phosphoflow class where we did simple 2-parameter stuff. Now we don't. Now it's like straight into PBMC. But here's the classic kind of cell line experiment in the U937s that we always recommend people to try if their experiments aren't working, where you can induce with interferon gamma to induce STAT1 or IL-4 to induce STAT6, and you can put the combination in, I don't have it here, to actually induce kind of both of those phosphoproteins. And so that's a nice simple experiment to run, and just lets you know that you're doing the technique right. So moving from cell lines to primary cells, normally we actually spent kind of a whole lecture on doing surface markers, and I'll just talk about that kind of briefly here. And then understanding your stem, I think this is really important for people, especially transitioning from kind of cell lines to primary cells, and what to expect there, that all your stems don't necessarily work the way that they do in cell lines. And this, I think, is a really important point that you kind of learn as you start to do this technique more. So we basically did some experiments where we did signaling responses in vivo and in vitro in mice. So in vivo, we actually injected cytokines directly in the mouse and then harvested the spleen rapidly and fixed it very rapidly as well. It took us about a minute and a half or so to get it out and fix it. And then we did phosphoflow. So for defining the cell subsets, these are a couple of the mouse surface markers, B cells and T cells with B220 and TCR. And then CD11B demarcates kind of your neutrophils, and then more in the middle, your macrophages, DC, NK. There's a bunch of stuff in this sort of CD11B intermediate zone. But just to kind of touch on a couple things, one of the keys that I kind of mentioned before too for doing the surface staining is, number one, the titration that you typically use on live cells probably is not the best titration to use on your fixed and perm cells. And I would say that's probably the case 95% of the time. So almost always I've seen you want to titrate is you're going to actually use less antibody than what's sort of recommended by the vendors. The vendors always hate it when I say that, but a lot of the time these antibodies like B220 or something, I don't even know what's recommended, but we'll use these things that might be recommended at 1 to 100 or something like that, depending on your color. We'll use that at 1 to 1,000, 1 to 2,000, something like that. So you might actually use about a tenth or a 100th sometimes of what they actually recommend. And the other thing too is that depending on the color or the conjugate, you might have a lot more or less background binding too. So once the cells are permeabilized, some of these tandem dyes, Cy7, things like that, might get a little bit more sticky. And so these guys, you tend to see sometimes a little more background when you throw those in there. So, PE Cy7, which might be just a blazing stain on your live cells and give you, 100-fold signal to noise. All of a sudden you throw that in on your perm cells and your background is 10x higher, your positives are 5x lower, and you can barely separate your cells apart. And so that's a case where I'd always recommend, titrate that antibody. Typically your positives will stay where they are, and that negative signal, that kind of that background binding, will decrease. So it's really, really, really important. I can't emphasize that enough to really titrate those reagents. So I guarantee you the first time you throw on your surface panel, it's going to look horrible. You're just going to have a big blob of cells, and that's the first thing that always happens to people. They have a big blob, and then they say, phosphoflow stinks, I'm never going to run this again. And so then they come here, they see that if you titrate it right, you get a beautiful surface stain, everything looks good, and they say, phosphoflow is awesome, I'm going to use it every day of my life. So really, you really have to test out those surface stains and really optimize those. And so I always recommend that's the first thing you want to do. Just live cell stain versus fixed perm cell, no phospho antibodies at all. Just really get that surface marker staining looking like you want it to. And make sure that your surface markers still give you the populations that you want them to, right? You don't want to, all of a sudden you have some subset that's 1% on live cells, and then you perm it, and all of a sudden you have 8% or something like that. there is that problem a lot where these antibodies will bind kind of to false epitopes or things inside the cells or whatever. So you want to make sure that you're still getting the right fraction of cells and then kind of the right distribution of cells as well. Yeah?
When you tried the different fixed upfront conditions, were there ones that were better for background staining or specific?
Yeah, so there definitely are. So if you do some of the more kind of detergent-based permeabilizations like the saponin, perm buffer 1 in the BD lingo, that actually is typically a lot better for surface staining. So the methanol seems to kind of bring out more of the badness, I would say, in the samples and more kind of background. So methanol is kind of the best and is required for some of the phosphoproteins, but is kind of the worst for some of the surface stains. And so this is the classic, what I was talking about, kind of the yin and yang of it, where it's something gets better, something gets worse, kind of thing. So a lot of the time your phosphos get a little worse, but your surface gets better, and that's just one of those balances that you have to take. Yeah.
Do you do any type of blocking before you come in and do the staining? And also, have you looked to see whether these are still just surface epitopes, but now that you've been, can you actually see it internally, some of those whole surface?
Yeah, that's definitely kind of an issue, right? I don't know if it's a problem per se, but if you have something that, is intracellularly expressed sometimes or sitting in endosomes or whatnot that might, come out to the surface, you definitely can see those. And so you might see a higher fraction. Again, that's what I was talking about with kind of the fraction of cells that are positive, where you can see those cells kind of all of a sudden now seeing that. The blocking agents, for the most part, blocking doesn't really do a whole lot to change any of these distributions. We've tried that quite a bit, and there really just wasn't a whole lot that kind of changed with that. There might be a couple antibodies that get a little bit cleaner, but for the most part, you can try to block with everything you want. I think there's just too much stuff inside the cell to try to throw some kind of blocking, and even if the BSA and the FBS and these things don't really block it out all the way, there's just too many kind of epitopes, too many charged things inside the cells to really block it out all the way, and we've tried everything and it just never got that clean. So if you find something, let us know to clean that out. I'm just going to go ahead and jump a little bit to the next one here so that Sean can get up here. A couple of things just to kind of point out. Choosing the right dose of the stimulus, this might seem somewhat obvious, but obviously sometimes different cell subsets might respond to different doses of particular cytokines. In this case, we have kind of the monocyte population and the B cell population. You can see they actually differed, a decent amount kind of in this sort of intermediate zone of IL-10 dose response, but for the most part, these stims are pretty comparable. Different stims might not be this way, so if you're looking at different cytokines, it's important to know that if you put in kind of a saturating amount of a cytokine, it might be really, really saturating for one pathway and not so saturating for another one, or one cell type and versus another cell type. So that's just something to be aware of when you're kind of profiling diseases and things like that you might really be super stimulating your samples and missing some of the heterogeneity. So it's worthwhile sometimes to do, kind of a high dose, a mid dose, a low dose, or something like that, and you might actually be able to see some differences in your samples if you're trying to induce them to do something in response to external stimuli. And at the same kind of token, too, the time point of stimulation is really important. So here is just an example kind of comparing in vivo versus in vitro, where we did LPS stimulation and kind of seeing in vivo, we had a really rapid time course from 0 to 5 minutes, 15 and back down. Here was 0 to 5 and back down, whereas in vitro that signaling actually took a little bit longer, more in that 15 to 30 minute kind of ballpark. So this is probably due to distribution and kinetics of distribution and things like that. But overall, this just kind of highlights the importance of just the kinetics of what you're looking at. In this particular case, if you had looked at 15 or 30 minutes, you would see nothing, and you would determine that nothing happened. So it's important, to try different doses. It's important to try different time points to make sure that you don't miss it. If you do half an hour all the time, you might just miss half of your signaling pathways. They might all be done already. And different surface markers can resolve different signaling pathways. So here's a case of where CD25, which is the high affinity IL-2 receptor, correlates really nicely with STAT5 as we put in different doses of IL-2 here going across the top. You can see these different populations basically popping out, and then kind of the ones that are IL-2 or CD25 low start to pop out at high doses, as you'd expect, just from kind of the low affinity receptor being about 100x worse affinity than the high affinity version. So you can just kind of see these sorts of things. If you have cytokine receptors now, IL-7 receptor is a big one that people are measuring for Tregs and things like that. You can correlate kind of those sorts of markers to the actual pathway, which you would hope and you would expect kind of that those things go together. So in the back, I just have some references for you guys to look at, some of the papers that we used here and stuff. So I'm not really going to go into these. That you can look up, and just a couple of really quick notes. Again, for your stimulus, always try to make sure that your stimuli are functional. In some kind of assay, be it in cell line or something, just to be sure that you have something working there. Keep your cells healthy. This is an important thing. Again, don't assume all the surface markers work, but also down here, don't assume all the cell subsets are represented equally, and also don't assume that primary cells are the same as cell lines, and that I think is an important thing that comes up a lot too. Just because your cell line that's a B cell line responds a certain way to a stim, it doesn't mean that your primary cells are going to respond that way, and that comes up a lot too. We get a lot of questions. This thing works great. How come it doesn't work on primary cells? And it just doesn't, and that's the way it is. So, cell lines are cell lines for a reason. They have a lot of messed up signaling pathways, and so you can get a lot of things that aren't kind of normal. And then I have some tips for just validating phosphoantibodies in the back. And I just want to thank this way back when Matt Hill, who did some of the work with me, Gary, and BD has always worked with us to developing a lot of the reagents, and they helped us along the way on these early experiments. So with that, if there's any other questions, thank you. Sure.
So, are there particular markers or kinds of markers which don't work after fixing perm on the surface?
Yeah, are there markers that don't work after fix and perm? There definitely are a lot of markers that don't work after fix and perm. So that's a pretty common problem, and is one that you just really have to just try to solve by trying more reagents in essence, and there's no real easy solution to that other than trying them, and also talking to the vendors, about what they've tried in these different fixed perm conditions as well, because they have tried a lot of these things, and so you might be able to find use their tips basically for getting the right antibody for the right situation for that. So sometimes though you just have to try different clones, and that's part of the business kind of on that. Yeah.
Is the common protocol to do surface staining fixed and then another stain, or is it to do all the antibodies together?
So you can do you can do kind of, that's what we call kind of a sequential staining technique, where you can do a surface or you can fix and do a surface and then perm. The only drawback on that is that certain fluorophores, if you're using methanol, won't be able to withstand the methanol. So PE and per-CP and these protein fluorophores actually get denatured in that process. So you can't come in with methanol basically after those things are there. You can use that for small molecule dyes, though, the Alexa dyes and Pacific Blue and these kinds of things. Those are stable to the methanol. So typically, if there's some surface markers that won't work after perm, we'll stain them with some of these small molecule dyes, then perm, and then come in with the other antibodies at the end of the method. And so that's one way to get these things that don't work to actually work.
How do you validate antibody, or which way? Validate antibodies for
The surface markers or the?
surface marker?
So typically the way that we validate is again, do a live cell stain, get your distribution of that, do the fixed perm on the cells too, and then basically make sure that distribution is the same distribution that you had in the live cell stain, and that's your gold standard, is whatever the live cell stain yields is what you really want to be seeing on the perm cell. And again, with the caveat that sometimes there's antigen inside the cell that you might also be staining.
And sorry, and for the protein inside cells, how you can be sure that your antibodies recognize the protein?
That's the $1 million question. You can't be. That's the challenge. Yeah, and that's where you. I had 1280, 1024, 60 hertz. Yeah, that's it. Worked okay. You really that's a hard question. I mean, that's just one where again with some of the actual signaling molecules, it's a little bit easier almost because you can see it get induced. And you can kind of correlate that to like a Western blot or something like that. If you actually want to look at total protein, it gets very difficult because it's not as easy to control for that. You have to use siRNA or overexpression or something like this to actually tell that. Yeah. Yeah, one last one. Yeah, you can do live dead stains. You just have to use kind of the fixable live dead stains, which is basically just kind of a covalent dye that binds to the cells. And so Invitrogen or Life Tech has some, and I'm sure other people have it too. I forget what they call it, something like fixable live dead or something. I could find that for you.
Aqua.
Yeah, there's aqua and other colors too. I forget what they're called though. Yeah.
So in those cases, you put the fix down, then put the live dead?
No, you want to do the live dead before you fix it. Typically, is the better way to go. So you actually want to put that in there, kind of as the cells are alive.
But you would want to stop the stimulation condition prior to get the phospho signal, right? So
Yeah, normally
somewhere between that 5-minute stimulation.
Exactly. Yeah, you would want to throw the live dead stain on and then do the fix basically after that. And you can dial the live dead too if you titrate it right, probably to where you can throw it in kind of with the fix, and it'll probably you'll be able to tell them apart okay too. You just need to kind of titrate that a little bit on that.
We've also done for like a lot of frozen primary tissues. There's a rest period after you thaw it out. Like even today, you guys have seen the protocol for BMCs. There's a rest period, and so sometimes we'll actually do the viability right when we thaw it out, and then wash them and let them rest post viability staining with the covalent dyes because once they're labeled.